Most of the data in ImmunoLand are focused on comparisons. Omicssoft curators carefully annotate each study found in ImmunoLand, utilizing controlled vocabulary. This controlled vocabulary allows comparison across multiple studies in ImmunoLand. Following curation, study data are analyzed using the comparisons identified within each study and are then deposited into ImmunoLand. An overview of the data available for each comparison can be found under Select View, Comparisons. Here, you can see the different categories of comparisons organized by tissue. Let's change the histogram to categorize by comparison category instead of by tissue. This shows that most comparisons in ImmunoLand are treatment versus control and disease versus normal. We can now group histogram columns by sample data mode. This shows that most data in ImmunoLand are microarray experiments. To get a better look at the types of comparisons available in ImmunoLand, let's use the gene SERPINB7 as an example. The default view for gene-level data is disease versus normal. In this view, each ImmunoLand comparison between disease sample gene expression and normal controls is shown as a different circle, with change in expression plotted on the x-axis and statistical significance in the difference plotted as the circle size. Selecting on a circle will show additional details about the comparisons. The disease 1 versus disease 2 view will only show comparisons between two different disease samples, grouped by disease category. The cell type 1 versus cell type 2 view will show comparisons between cell types. Treatment versus control will show any comparison where a single cell type is being compared in response to a treatment, such as to a drug, cytokine, or allergen. In this study, interferon treatment with LPS increases SERPINB7 expression compared to LPS alone. Comparisons between two treatments can also be viewed. Let's change the view so that we can see both treatments being compared. First, click Specify Multiple Profile Columns. Now add Control Treatment. We also have a view for responders versus non-responders and comparisons between tissues. There are also a few specialized comparisons that can be viewed under Other Comparisons, such as inflamed versus non-inflamed. Since each ImmunoLand comparison is generally comparing expression of thousands of genes, we can look directly at those expression changes at the comparison level. To do this, we can search for an ImmunoLand comparison title and enter it in the search box. The default view for comparison level data is a volcano plot of all tested genes in the comparison. We can quickly find the most affected genes in a given comparison. On the x-axis, the log2 fold change estimate is shown, and on the y-axis, raw p-value is shown. We can change to adjusted p-value in Specify Columns. We can also add cutoff lines. Selecting a group of genes will automatically label them. We can also look at gene expression changes across comparisons. Let's go back to SERPINB7 in the disease versus normal view. Let's filter for disease state psoriasis. Now select several of the studies. Then click Browse Selected Comparisons. For each disease versus normal comparison within the psoriasis disease state, we have a separate volcano plot. Let's select some of the upregulated genes in the first plot. You will see the same genes selected in the other plots. Notice that some genes are consistently upregulated in lesional versus non-lesional samples. Another useful view at the comparisons level is the Venn diagram. For each set of 4 comparisons, we can see genes in common between significant gene sets. Cutoffs can be changed under the Cutoff tab. Additional comparison level views include heat maps and pairwise correlation, and additional comparison analysis tools such as principal component analysis, summary statistics, and finding significant genes and pathways within these comparisons are available. For example, the significant genes view displays the full list of all genes, with the number of selected comparisons in which each gene's expression is significantly different. Let's sort on the number of comparisons in which these genes were found to be upregulated. We can see that a number of genes are consistently upregulated in these comparisons. Let's make a gene set from these. Click Create Gene Set, create gene set from selection, and name it. Now we can search for these genes by the gene set name. This heat map shows each disease versus normal comparison with the relative expression for each of the genes we selected. Let's filter for disease category, skin, and connective tissue. This shows that our set of genes is consistently upregulated in skin and connective tissue disease.